whether it's a rearrangement of c-MYC to kappa and lambda. And that we see in about 12% of our cases in Singapore. So it does happen from time to time, not that common, but you do that. Then we come to this problem. Is there such a thing as MYC-negative Burkitt lymphoma? Look at this case, at a starry sky appearance, quite uniform cell morphology, can go with Burkitt lymphoma. The immunophenotype is okay for Burkitt lymphoma, typical kind of a 100% Ki67, CD10 is positive, BCL2 is negative. However, when you fish for it, there is no MYC translocation. So no break apart of c-MYC or IgH, kappa, lambda, all normal. So no translocation of c-MYC to any of the immunoglobulin genes. By right, you cannot call it Burkitt lymphoma. But then if you look at it, it looks like Burkitt lymphoma, it behaves like Burkitt lymphoma. It has to be. So what are you going to call it? MYC-negative Burkitt lymphoma. Do they exist? Well, increasingly we recognize that they do. Now, what is the reason for this? There are a few possibilities. The first is that there is a breakpoint, but there's a translocation that's been missed by the FISH probes currently. It's possible that the breakpoints are too far away, and therefore the current FISH probes fail, the break-apart probes may fail to detect them. So that's one reason. Second reason is that the c-MYC could have been inserted into an immunoglobulin gene. Let me show you an example. This is a case where you can see that using a break-apart probe, there's no translocation, no rearrangement of c-MYC. IgH looks intact. Dual fusion, we don't see it, but if you look at IgL, by break apart, there is actually a break apart of IgL. What has happened is that c-MYC got inserted into the IgL gene, and because of that insertion, the effect is the same. There's overexpression of c-MYC, and the c-MYC protein actually is overexpressed. So sometimes this can happen. So it's still a Burkitt lymphoma, but the immunoglobulin gene has broken apart, and c-MYC got inserted into the immunoglobulin gene. There is another possibility, and that is dysregulation of microRNAs, and there have been some publications about that. The idea is that instead of a translocation resulting in overexpression of c-MYC, which drives the disease, what you have is dysregulations of the microRNA, which results in overexpression of c-MYC. And when you can, in these cases, when you stay for c-MYC, c-MYC is still highly expressed even though there is no translocation. So that is another possibility. And yet, there are other reasons. And one of those other reasons is now appearing in the WHO classification, the updated WHO classification. This is a new entity, Burkitt-like lymphoma with 11q aberrations. And what you have is a subset of lymphomas that they resemble Burkitt. They look like Burkitt by morphology and immunophenotype, but there's no c-MYC rearrangements. Instead, they have alterations of 11q. And the alterations are quite interesting. They have the proximal part, 11q23 is amplified, whereas 11q24, right to the terminus, is actually lost. And this is going to be quite challenging to detect this alteration. These cases tend to occur in the lymph nodes, tend to be younger patients. Morphology-wise, they're a little bit more pleomorphic. The experience is rather limited, but so far these cases tend to behave quite similar to Burkitt lymphoma. So for the present moment, they are called Burkitt-like lymphoma with 11q aberrations. This is taken from a publication, and you can see that the immunophenotype and the morphology is similar to Burkitt lymphoma, but they do not have MYC translocations. The karyotype is a bit more complex, but these are the minor alterations. And how do you detect this? You can use CGH. By array CGH, you can see that the proximal part 11q23, there's amplifications, whereas 11q24 to the terminus, there is loss. So potentially what you can do is to design a FISH probe around this area which should be amplified, whereas design a FISH probe targeting this area which will be deleted. So that's potentially something we can do to detect this particular alteration at this new entity. Now, as we move towards the end, I want to talk about high-grade B-cell lymphoma. There are two categories of high-grade B-cell lymphoma in the updated WHO classification. The first is what we call the double and triple hit lymphomas, which now call high-grade B-cell lymphoma with c-MYC and BCL2 and or BCL6 rearrangements. Now, in the past, many of these were called the gray zone lymphoma with features intermediate between DLBCL and Burkitt lymphoma. And in the past, if you get a case that looks like DLBCL by morphology, very pleomorphic and so on. But if it does have the same double hit translocation, we'll call it DLBCL with a double hit translocation. We still call it DLBCL. Now, with the updated classification, we include all of them under the same category. As long as it has got this double or triple hit translocations, they are grouped together as high-grade B-cell lymphoma with MYC and BCL2 and or BCL6 rearrangements, regardless of the morphology. So some of these cases were currently Burkitt with double triple hit, which is not Burkitt. Some of these were called DLBCL with double or triple hit. Some of these were called gray zone lymphoma, all now grouped together by the genetic alterations. So what does this mean? It means to say that if you do not perform FISH, we cannot diagnose this entity. So you will always miss this entity unless you perform FISH. Then the other group of high-grade B-cell lymphoma are cases that have features that look intermediate between DLBCL and Burkitt lymphoma, but when you FISH them, they do not have this double hit translocation. So what are you going to call them? You call them high-grade B-cell lymphoma, NOS. Let me show you some examples to illustrate that. So the first group, these are aggressive, the double triple hit lymphomas. These are aggressive B-cell lymphomas with rearrangement of c-MYC and BCL2 or BCL6. In some cases have both, there's a triple hit. And they exclude cases with other double hit translocations. The BCL2 and BCL6, you don't call them. So this is referring to c-MYC with BCL2 and BCL6. If you've got c-MYC and cyclin D1, again, you don't consider that. So some cases of mantle cell lymphoma with cyclin D1 IgA translocation also have MYC translocation on top. You don't call that in this category. You call them still a mantle cell lymphoma. Usually they tend to be blastoid variants. Okay, so they include some of these categories which we currently call, as I mentioned. And I'll show you an example. This is a case which was previously diagnosed as gray zone lymphoma with features intermediate. And you can see why. Not terribly starry sky, but the chromatin is quite evenly dispersed. There's about they're about uniform looking, not really big nucleoli. So can go for Burkitt lymphoma. But if you look at the KI67, it's not quite 100%. CD10 is there, but look at the BCL2, far too positive, too strong. So we call it gray zone between the two. This case has the rearrangement of c-MYC, rearrangement of IgH, BCL2, as well as BCL6. So it's a triple hit lymphoma, rearrangement of all three. This will go into this particular category of high-grade B-cell lymphoma with c-MYC and BCL2 and BCL6 rearrangements. Now, this is another case. This is another high-grade B-cell lymphoma with some overlapping features, some starry sky appearance, quite uniform-looking tumor lymphoid cells. Morphology can go with Burkitt lymphoma. Immunophenotype, CD10 is there, BCL6 is there, but BCL2 is too positive, even though KI67 is wall-to-wall. So with this, again, c-MYC is very strongly stained, suggesting there's a MYC translocation. But because BCL2 is so strong, therefore you're wondering this could be a double hit. Very likely a double hit between with c-MYC and BCL2. But when you FISH, there's a c-MYC and IgH, BCL2 is intact. There's no rearrangement of BCL2 nor BCL6. So you can't call this that's a group of a lymphoma with c-MYC with a double hit and triple hit, c-MYC with BCL2 and c-MYC. So you have to call these cases, oh no, I keep pressing the wrong one. So you have to call this high-grade B-cell lymphoma NOS in the new updated classification. So that's how we use that. So I think these are a group of conditions that we probably will need molecular techniques, particular FISH. I had a discussion earlier with Prof. Koh there. I think that if you try to use next-gen sequencing and so on, it will not be that useful because it will be very difficult to know where the breakpoints exactly are in every case. So FISH is still the better way of detecting translocations, and that's probably what we may need to do. The last bit I'm going to mention very quick as I end is that of lymphoma diagnosis that's supported by molecular findings. And I think I'd like to mention a little bit about CLL here because we know how to diagnose CLL. The only alteration apart from the numerical criteria is the fact that you need definitive disease, needs persistent disease for at least 3 months now. But the diagnosis of CLL or SLL is not difficult. We know this disease. You can see in the small lymphocytic lymphoma, these diffuse infiltrate lymphocytes with these pale proliferation centers, which are composed of pro-lymphocytes with a more conspicuous nuclei in these proliferation centers. And immunophenotype, again, we all know CD10-20 is positive, and this is a B-cell lymphoma that shows aberrant CD5 expression, positive for CD23 and negative for cyclin D1. So no problem diagnosing CLL in most cases. In some cases where, because the CD23 is not so positive, the CD5 is a little bit weak, you have a bit of difficulty, now we have this marker called LEF1, which is a transcription factor that you see in CLL or SLL and not in many of the other low-grade B-cell lymphomas. This can be helpful in the diagnosis again. So, do you really need molecular techniques? Not for the diagnosis most of the time, but we do need it in terms of treatment. Because as you know, the CLL and SLL, this is not a uniform disease, it's a heterogeneous disease. They've got cases which behave more aggressively, there are cases which are more indolent. You can predict the behavior by looking at some of these cytogenetic alterations. This forms what we call the CLL panel. If you have got a deletion of 13q alone without any other alteration, this involves the microRNA. If this is the only alteration you see, and this alteration is seen early in the disease, one of the earliest sign of the disease, then they have a very, very good prognosis. You can tell a patient to go back home, you don't have to worry too much about it. The patient will probably, elderly person may die from other diseases from the heart rather than on CLL. Very good prognosis. On the other hand, if this patient has got a deletion of 17p involving p53, this is poor prognosis. And one of the treatment that's been commonly given to these patients, CLL, contains fludarabine. And you know that fludarabine-based therapy will not work for these patients with this 17p deletion. So these patients do not work very well with the standard treatment. So they need a different kind of treatment. So therefore, there is a need from the treatment perspective to know which patient should be given what kind of treatment, and therefore molecular techniques may be useful. So although it's not necessary for classification, it is particularly useful for prediction of response to therapy. And over the years now, we know that the number of mutated genes are present. Many of them involve the B-cell receptor signaling pathway. And these mutations have prognostic implications, but also have predictive implications. So this is the chart just showing you the various the pathways which are operative in CLL, as well as the number of drugs that target different parts of these pathways. So looking at the mutations that are present in CLL and SLL may be useful in predicting what kind of treatment the clinicians will use, particularly for relapsed disease. So for that reason, going forward in the future, we may have to do more than just classify the disease, but we have to predict what drugs may be useful, and therefore molecular techniques, even in CLL, may become necessary. But as I end, I will say that because there are quite a few genes that are operative here, trying to do Sanger sequencing on each one of them is probably a no-go. So going forward to the future, probably we will need targeted next-gen sequencing to look at these various pathways and alterations that may be operative, so that clinicians can choose the best drug to treat these patients. So with that, I thank you very much. So I will end off with this last slide, sorry. That's lymphoma classification in the molecular age. Diagnosis is still based on the clinical findings, the morphology, immunophenotype, and the genetic and molecular findings. None of these by themselves is the gold standard. You probably need to involve all of them in the making of a diagnosis. Nevertheless, there are now a number of entities which are heavily defined based on the molecular alterations. And therefore, to classify some of these newer entities, you will need greater use of FISH in particular, and in some cases, sequencing in some of these difficult cases as well. Going forward, peering through the looking glass into the future, I believe that targeted NGS will play an increasingly important position in the diagnosis, the prognostication, and in particular, the predicting the response to therapy. And this is something that we as pathologists should embrace. We should not be afraid of them. We should embrace them, and because if we don't make use of this technique, our clinicians will do it and drive us out of a job. So with that, thank you very much.